Myeloma research continues at an incredible pace. Questions are being asked like, should we treat high-risk smoldering myeloma with new immunotherapies? What is the best frontline therapy for multiple myeloma? Can we use bispecific antibodies by themselves in early relapse? These and more questions are the ones that myeloma researchers are trying to answer and are answering, and have been a huge part of the recent major conferences of ASCO and EHA. ASCO stands for the American Society of Clinical Oncology, and EHA is the European Hematologic Association. In addition to that, we had our own International Myeloma Working Group Summit in Stockholm in June. I'm going to take the best of those meetings that relate to multiple myeloma and share them with you today so that you and those you love can learn more about what's happening at the cutting edge of research in multiple myeloma. Hi everybody, Dr. Joe here from the International Myeloma Foundation, and it's my privilege today to give you a quick review of the major updates that came to us from ASCO, EHA, and the IMWG workshop. Just before I start, let me remind you to subscribe to our YouTube channel. When you do that, you'll be kept up to date and you'll help others be kept up to date in the latest and greatest in multiple myeloma. All right, let's dive in. I'm going to start with 3 very important clinical trials. Easy to remember, 3 trials because they were all phase 3 trials. You may remember that phase 3 clinical trials are really the highest form of evidence. This is when we compare a new strategy to the standard of care. Phase 3 trial number 1 was called Monumental 3. This was actually highlighted in the top abstracts of the EHA meeting. And this was a study that was bringing in talquetamab, a bispecific antibody that we typically use in late relapse disease, and bringing it earlier in combination. Just as a quick review, bispecific antibodies are drugs that grab onto the myeloma cell and also to a local T cell to engage it to help destroy the multiple myeloma. What's unique about talquetamab is that it targets a unique target on the cell of myeloma called GPRC5D. And so we can use it now in monotherapy or by itself in very late relapse after 4 prior lines of therapy. But in this Monumental 3 study, they brought it into earlier relapse and they combined it with daratumumab plus or minus pomalidomide and compared it to what we would typically do in early relapse to use a combination like we call DPD or daratumumab, pomalidomide, and dex. Well, the results were striking. Whether it was the talquetamab plus daratumumab plus pom or just talquetamab and daratumumab, both of those arms did so much better than the DPD arm. In fact, we saw that at the 3-year time frame, over 80% of these patients were still in remission, compared to 30% in those patients with the usual triplet. I think this study is really important because we're going to see a further move of bringing bispecific antibodies, not just in late relapse, but bringing them earlier into relapse. Not only so, bringing them in combination so that we can get a deeper and more durable response. There were side effects, of course, that we're still learning to minimize with the use of talquetamab, but we saw considerably fewer infections with this combination than we've seen before. Phase 3 trial number 2 was also with a bispecific antibody, also used in earlier relapse, and it's called the Majestic 9 trial. The Majestic 9 trial took teclistamab, another bispecific antibody that targets BCMA, and now used it by itself and compared it to the usual drugs that we use in early relapse. This is a follow-up to a study that you may have heard me talk about last year after our annual American Society of Hematology meeting, where teclistamab was partnered with daratumumab. And some would say, well, by the time we get to relapse, some patients have already had daratumumab. So they did this study to look at teclistamab alone. And again, the results were impressive. Here, at 18 months, we saw that 70% of patients were still in remission, much, much better than those patients that had the triplet or the doublet that it was compared to. I think this will be particularly important. We anticipate that, just like I mentioned with the talquetamab study, that these two combinations, these two drugs in this format, will likely be FDA approved in the near future for us to use earlier in the disease course so that we have more options for patients and we can bring the strength of these bispecific antibodies not only to late relapse, but also to early relapse. The final phase 3 clinical trial also has a great name. I love these names: monumental, majestic trial. Here we have the Successor 2 trial. And the Successor 2 trial introduces a new drug to us that we do not have yet FDA approved from a new class of drugs that are also yet to be FDA approved, although we hope to see them this year, called CELMoDs. CELMoDs are pills, they're oral drugs that are kind of like the next generation of immunomodulatory drugs. These two drugs in the CELMoD class are called iberdomide and mezigdomide. The mezigdomide, if you're a soccer fan, we sometimes call it Messi or Mezzi for short. But the mezigdomide was the one studied in the Successor 2 trial. And mezigdomide was combined with carfilzomib and dexamethasone and compared to carfilzomib and dexamethasone alone in relapsed multiple myeloma. Most patients had had 2 or 3 prior lines of therapy, but some patients had had even more lines of therapy than that. The results were also very impressive. Patients who had this novel combination with mezigdomide, carfilzomib, and dex on average stayed in remission for 18 months, whereas it was only 8 months with carfilzomib and dexamethasone alone. This will almost definitely lead to an approval of these CELMoDs, in particular mezigdomide, quite likely late in 2026, and give us yet another option to treat patients, especially an option that's easily accessible by being a pill that doesn't require people to come into the clinic so regularly. Well, there you have it. An amazing set of phase 3 clinical trials that really were the highlight of the myeloma abstracts at the American Society of Clinical Oncology and the European Hematologic Association. But don't run away yet, because I'm going to fill you in on some of the other amazing abstracts that will very much change the course of multiple myeloma in the months and years to come. Before I continue, I want to talk for a moment about clinical trials. I just shared with you 3 practice-changing phase 3 clinical trials. And the only reason why we have these great drugs and can bring them to the clinic is that patients with multiple myeloma have been on clinical trials. Sometimes we hear clinical trials and it may scare some people and think, oh, am I just going to be a guinea pig? Am I going to just receive a placebo? That's not how we do it in multiple myeloma. This is an opportunity to have earlier access to therapies that could likely change the way we treat multiple myeloma. We encourage you to learn more about clinical trials, to come to our IMF events where we discuss them, and even to search clinical trials that could be available to you. If you go to myeloma.org/SparkCures, we have a search engine built into our website that can help you find clinical trials close to you. As we approach closer and closer to the cure in multiple myeloma, clinical trials are going to be critical for us to provide that for our patients in the future. Well, let's get back to the science. In the introductory part of this review of ASCO and EHA, I shared with us 3 huge phase 3 clinical trials. Well, let me tell you about some other very influential studies that are very likely going to change the way we think and treat myeloma. Several trials are looking at smoldering myeloma, that phase of myeloma before it becomes true active myeloma, using bispecific antibodies, in particular elranatamab and teclistamab and linvoseltamab. These drugs are being studied to see if can we intervene early enough to catch this disease such that it never becomes multiple myeloma. And some of those preliminary studies are being done, and I think will change the way we think about high-risk smoldering multiple myeloma. In the frontline setting, we saw several studies bringing drugs that we've historically used later in the disease course to the front line. One in particular I'd like to highlight is Belantamab mafodotin. You may remember that Belantamab mafodotin, sometimes called Bela, is an antibody drug conjugate. So it's a drug that hooks onto a myeloma cell like antibodies do, but conjugated or stuck to it is a drug, a toxin, that it drops into the cell to help destroy it. It's now approved in relapsed myeloma, But several studies were presented where it was brought into the frontline, adding it to VRD or Velcade Revlimid dexamethasone or RD Revlimid dexamethasone, the combinations we often use. We're still sorting out the exact dosing of this drug, but I think it's something we're going to be able to offer patients in the frontline setting in the near future. Furthermore, looking to the future, I was very excited to see the number of novel ways of treating multiple myeloma that are being developed. Let's take CAR T, for example. CAR T-cell therapy has changed the way we treat myeloma, and many patients are living longer and better now because of it. But can we make even better CAR T-cell therapy? One of them is now having a new target. Right now, the two CAR T-cell therapies we use target something on the outside of the myeloma cell called BCMA, but now we have a new target, GPRC5D, and we saw updates from a drug called Arlo-cel that is being developed to target that part of the myeloma cell. We know CAR T-cell therapy is great, but most patients sadly still do relapse later, and having another CAR T that can target the cell differently will be really important. Furthermore, we saw an update of the so-called in vivo CAR T. What does that mean? Well, right now, when we do CAR T-cell therapy, we take T-cells out of a patient, we train them to know what their myeloma looks like, or we treat them so that they have a receptor on their outside that is specific to the multiple myeloma, then we give them back to the patient. That takes time, it takes manufacturing, and it does require some extra chemotherapy to be given to the patient before those T-cells are given back. In vivo CAR T means that we actually directly give patients a drug and it goes to their own T cells and converts them to CAR T cells. Amazing. We saw this reported last year with only 3 patients. Now we have 18 patients reported. Incredibly, all 18 achieved MRD negativity in the first month. So this is a remarkable therapy, not only because it's very effective, although the numbers are small and we need to validate this in larger numbers, but it was also safer in the sense that we saw fewer side effects than we often see with CAR T-cell therapy. Not to mention, this could make the science of CAR T-cell therapy even more accessible to patients in the future. And then I'd like to highlight what's new in bispecific antibodies. We already have 4 bispecific antibodies approved in multiple myeloma, and that's wonderful, but there are more coming. One of them in particular is called Etentamig. And Etentamig is a little bit different than the other bispecific antibodies because from the very start, it's given once every 4 weeks. Most of our bispecific antibodies start weekly and then maybe we taper it down to every other week or once a month. But here it's given right off the bat once a month, and we see less of the early side effects that we typically see with bispecific antibodies, specifically the cytokine release syndrome. In fact, in their study, they showed 2 interesting things. One, they could give this drug after someone had had CAR T-cell therapy, which gives us more options after CAR T. And interestingly, when they gave what we call prophylactic tocilizumab, that's a mouthful, but that just means we give a drug before we give the bispecific antibody to prevent a patient developing CRS, that actually 0% of patients developed cytokine release syndrome. This could make this drug much more easily given out in the community and not necessarily in an academic center where most of our treatments are being given now. So this is something not yet FDA approved, but I think is a little view into the future of what we're going to be able to do in multiple myeloma. There's so much more that I can say. Other things that are coming in the future, novel bispecifics, new antibodies, new small molecules, even something called Allo-CAR T, where we collect T cells from healthy donors to give to patients. All of these are part of the amazing research that's going on in multiple myeloma that will inevitably help our patients live longer with this disease, live better with the disease, and bring us closer to the cure. Well, these are exciting times in multiple myeloma with so many things going on, but please feel free to reach out to us. Subscribe to the YouTube channel that you see below you. If you want to speak to someone, call our info line or come to one of our events, whether they're in person or virtual, and you can learn more about them at events.myeloma.org. By the way, if you want to dive even deeper into what's going on in myeloma research, then you may want to check out a webinar that the IMF just hosted. The three doctors on our board, Dr. Vincent Rajkumar, Dr. Sagar Lonial, and Dr. Nikhil Munshi, shared their thoughts about what was happening in multiple myeloma, moderated by our own Heather Cooper-Ortner. You can just click the link here and check it out, and you'll learn even more about what's happening in multiple myeloma. I hope you enjoy it.